Thank you very much. From Bangladesh, actually, it's good afternoon, I should say, to you. So I'm very pleased to invite me as a speaker of this August gathering. Instructions and requested regarding the webinar Korea and Southeast Asia's efforts to achieve the UN SDGs. So respected moderator, delegates, scientists, ladies and gentlemen, I'd like to prepare my slides for actually what are the crop varieties developed in Bangladesh so far through induced mutation. That's why I make the title is crop improvement through induced mutation, combating food security. So if you see the plant mutation breeding and also the plant breeding, this process actually started, from the ancient times. And basically it is our focus on actually to create the variation. So we are actually altering the genes or genetics of the plants to create the desirable traits. And we have to involve the selection or creation of genetic variation and phenotypic evaluation of plants. And selective pressure by humans, we have seen that the seed size, seed shattering, plant height, actually is manipulated by the human being. So why is plant breeding needed? So historically, you see the domestication and the evolution of the germplasm or genotypes of the plant species, and also the breedings and in the foundations for modeling the modern society. So presently, actually, the population is increasing day by day, and also it actually creates pressure on the food security. So many approaches actually we have approached for the food security, including plant breeding. So plant breeding actually basically, it's a cross breeding, just the cross between the male and female parents. That is the conventional breeding. We call it hybridization. And also we have now mutation breeding, where you have the very narrow genetic base or no variation. Then you have to proceed with the induced mutations, and then you can create a lot of variations, and you can select the right variety or right genotypes. Of course, we have also the transgenic breeding, like genetic engineering, where you can integrate the genes from the foreign sources into your commercial variety. So mutation breeding actually provides the genetic variation for the plant breeding, where the conventional breeding has a very little scope or no scope. Then we can pursue the mutations. So mutations just simply the heritable change in the genome, and the mutations are naturally occurring or can be induced. So the Hermann Muller, actually, he got Nobel Prize in Physiology or Medicine in 1946 for the novel variation in the cells through ionizing radiations. And also later then, the Stadler, also they use the X-rays in maize and wheat and barley, rice, and so on, and many crops. And later then, the many scientists approaches the induced mutations. So, so simply that's the process of plant cells with mutagens to facilitate the crop breeding. So we can irradiate even any organ like seed, pollen, cell culture, plants, meristems, tissues, any organs you can treat with the radiation of the chemical mutagens. So so basically, actually, induced mutations create the new variation where you not find in the nature, and generate the novel allele or traits of magnitude faster than the occurs spontaneous. and generate variation in organisms where traditional integration is impossible, where linkage drag and asexual propagation. So it is broadly applicable in the field of crop improvement. So we have actually two types of induced mutations, that is physical and chemical mutagens. Physical mutagens just like X-ray, gamma ray, beta ray, these are the physical mutagens, and chemical mutagens, like EMS, ethyl methane sulfonate, sodium azide, and so on. So where you can change in the genomes at the gene level or chromosome level or genome level in nuclear and cytoplasmic DNA. And of course, you will have the recessive mutations will be expressed only in the homozygous conditions after self-fertilizations or shift mating of the mutated plant. This can occur in M2 generation following self-fertilization of the N1 plants. And single dominant gene mutations occur at very, very low frequency. So this is actually the schematic view of mutant generation. So just you have to apply your physical or chemical mutagens in the seeds. Then you will have that M1 plants, and in the M2 you'll find the lot of variations. I mean the segregating populations, and after that you have to select the right plants or lines for desirable traits. And of course, it follows the Mendelian segregation or Mendelian genetics, 1:2:1. So this is the simple, the mutation breeding, how it works. That's when you have the irradiation or the chemical mutations in the seed, you'll have M1, which is chimeric. So different types of plants you will find, and then you have to phenotype it, and then you have to select through the multi-location trial. You can alter the the many traits of the plants, whatever you like. You can change as you like in your what you desire. So this is the effect of the effect of the effect of the mutagenesis in the M1 plants, while you have find the variation. So you see that this is the examples of the clonal or cell lineage variation, and also the positional variation. So you can easily you can create the variation in your plants, like ornamental plants, bicolor or chimeric plants you can create. So you can have also the somatic mutation, like mutation in two types: the gametic mutation and somatic. Somatic mutation, you can change only the the somatic cell. I mean the surface of the plants, like petal color, flower color. You can change. So change in the sequence of the somatic cell, and any mutation that occurs in a cell other than a gamete, germ cell, or gametocyte. So this is the genomic mutation. There is aneuploidy, also chromosomal fusion, and you will have the changes in the chromosome, like duplication or translocation of the segments. And there are many spectrum of the mutations, like point mutations, insertion, deletion, duplication, translocation, and inversion in the chromosome. So basically, the structural and the numerical changes in the chromosomes, where you will find your desired plant types. So there is a lot of genetic concepts, like you will have the addition, deletion, substitution, duplication, and inversion. So many variation you can create through mutation. This is the example of the there are many types of the variation in the chromosomes. I'll not go details in that. So so basically the gene mutation creates in the nucleotide constitutions changes in the nucleotide constitution, like deletion or substitutions, like gene mutation in Drosophila melanogaster. So where you can change the the gene particular sequence you can change through induced mutation. You can have the silent mutation, missense mutation, nonsense, and prem-shift mutation. And in terms of gene action, a mutation may be recessive or dominant. Like recessive mutation to the recessive allele, and dominant mutation is the change to a dominant allele.
So we are short of time. So can you go to the conclusion?
cereals, flowers, legumes, fruits, vegetables, fibers, oil crops, and many tuber crops, and many other varieties of the villages. So now let's see the success stories in Bangladesh. So we have 124 varieties at Bina, but 85 out of 124, 85 are the mutant varieties we developed through the induced mutations. For the cereals, we have 26, like rice, wheat, and other cereals. And pulses, we have 40 variety legume crops. Wild seeds, we have 32, and horticulture crops 23, and fiber crops we have 3. So these are the common, the popular varieties. You see the Bina Dhan 7 for rice. Actually, we call it Dhan. Dhan means rice. So this is the early flowering, short duration varieties, and also it eradicate the hunger in the northern area in Bangladesh, where there is no employment, where there is no food of the people. So that is totally, your land will become fallow due to the short duration of the varieties. Now this land is become under cultivation. Again, actually, we have also drought-tolerant varieties. We irradiated the Nerica, new rice for Africa. We collected the seeds from Africa, Uganda, then we irradiated drought-tolerant. And also we have late potential rice variety where there is no crop in the field, and especially in 14. So it can be harvested, after after the regular season. So this is the pictorial view of the drought-tolerant variety in the hill areas, where the traditional people, the tribe people, actually they grow the varieties. So you will have the 25 to 31 ppm iron, and then the 27.5 ppm zinc. This is an unpolished rice where you can eradicate the malnutrition. So we have mainly about 20-30% people are malnourished. So we are now introducing these varieties to the the rural people of our in the farm areas and our farm community. So we have aromatic rice variety Bina Dhan 13 is aroma. We developed it through irradiation from the local variety. Also, we have the premium quality rice variety. Recently, we developed this long slender variety, and it is a very exportable variety in Bangladesh. Now we can export this variety. We have a wheat variety, salt-tolerant wheat variety. It can tolerate up to 10 to 12 ds per meter at the reproductive stage. We have mung bean varieties, that is legumes, mung bean. So the 80% fruit is shiny and it attracts the consumers. So consumer preference is very high. And we have the chickpea varieties that is called horse gram. This gram. We have 25% protein in this variety. It is tolerant to different diseases. About 92% edible oils we are exporting every year. So a lot of the revenue budget we are expanding from the governments. Now we are focusing. We are trying to reducing the the import. So now we have the very good variety. Also, sesame variety, it is used in the confectionery industry, and in the cakes, in the different food items, also the finger foods. So it is very good variety, and oil content is 52%. Also, we have the the beta and oxalate amino the Lathyrus variety. So we have the Lathyrus varieties. So we have black seed coated lentil variety. That is we use actually the indigenous mutagen through datura extract. So we have black seed coat. It is a very much good markers for the farmers. So farmers can easily identify this variety. We have lentil variety. Protein content is about 30%. And it is also high yield at 2 points. Usually the yield is 1 ton or 1.2 or maximum 1.5 tons in our country, but it yield is too much high, the outstanding yield. And we have the fiber variety. We call it vegetables. We can use it for vegetables, like jute variety. We developed through gamma irradiation. So what is the impact? Now let's see the impact. If you see the impact, so we eradicate the, the seasonal unemployment in the northwestern part of the country, and then it facilitated the increasing the cropping intensity. So now our cropping intensity is higher. We have salt tolerant rice varieties which cover about 30% of the coastal area. Also, and also the other crop varieties it area covers in the countries. And we have highly and climate resistant also a lot of varieties for sustainable nutrition security and helps in ending the hunger and sustainable relation to SDG goal 2, end hunger. So we have some future trust. We are looking the collaboration and joint program with IAEA developed countries and the universities and the CZ centers for,